It is my pleasure to introduce our second speaker, Martin Steger. So Martin did his PhD in molecular cancer research at the University of Zurich in Switzerland. He did his postdoc at the MPI of Biochemistry in Germany, working on Parkinson's disease and using global proteomics and phosphoproteomics to discover the first molecular marker for LRRK2 protein kinase. He is currently an industry researcher or head of Degrader biochemistry and co-founder at NEOsphere Biotechnologies, where he uses state-of-the-art proteomics methods for screening protein degraders and validating target candidates of degraders. And I will hand it over to you, Martin.
Pointer here. All right, so I will talk about proteomics and how we can use proteomics to discover targets of degraders and also use it for target validation. So Rebecca gave a very nice introduction about different modalities. In my intro, I would just like to showcase, maybe I have to remove this window here, showcase a few things about the active degrader programs currently pursued by pharma and biotechs. And according to a recent market report, those are 500 active such programs with about 70% involving molecular glue degraders or PROTACs. And if you look at the landscape of targets that are pursued by companies developing glues, it becomes evident that this is quite limited. Many of them are focusing on targets such as GSPT1 or IKZF1, 2, or 3. And although ligases other than cereblon and VHL are being explored for TPD purposes, those two remain the main ones, and with cereblon being the more versatile one, especially if it comes to glue applications. And looking at how many proteins have been disclosed as cereblon neosubstrates, according to a recent review, those are about 50 proteins. And so the question is really, are these proteins all, or is there more to discover? So of course, there are many research groups that have contributed to trying to answer this question with different essays. But I'd just like to focus on a few recently published highlights here. The first two is work from Monte Rosa, showing that about 1,600 proteins contain actually a cereblon-compatible beta-hairpin glue. These are predictions, and some of those have been nicely validated. Also, a more recent preprint in which was shown that the cereblon-dependent degradation of such neosubstrates is actually not restricted to G-loops, and this was nicely shown for WAF1 or also for G3BP2. And also, as shown by Rebecca, and also again work from Monte Rosa, interactomics approaches such as TurboID attacked with cereblon or in vitro affinity enrichment mass spectrometry with recombinant proteins identify hundreds of potential direct interactors of cereblon. Of course, in such assays, you see both direct and indirect interactors, but I believe that a good number of those are certainly direct interactors, as also shown by some of these groups. We took a little bit of a different approach. So we asked the question whether if we apply proteomics as a screening-based method with a larger compound collection, whether we could increase the number of neosubstrates that are actually degraded inside the cell. And having such large data sets at hand and testing different compounds, one can also ask questions about the connection between neosubstrate profiles and compound structure, and also find out how the biological context influences the degradation of neosubstrates. So here it's what I picked is our screening pipeline. So in this first study, we used 100 compounds that were confirmed to bind cereblon. These included both lenalidomide analogs and phenylglutaramides cereblon binding cores. We screened those compounds across 2 cell lines with complementary protein expression profiles. This works in a way that we do the treatment in 96 well plates. We use those plates for lysis and MS sample preparation. For the mass spec people among you, we use single shot, label-free, data-independent mass spectrometry. analyze the data, nominate candidates, and follow those up using mainly global ubiquidinomics and proximity labeling mass spectrometry. For the global ubiquidinomics, we use an approach called ubiquitin remnant or diglide peptide profiling, by which proteins that were formerly ubiquitinated are cleaved with trypsin, and this tryptic cleavage generates a so-called diglide tag on ubiquitinated proteins. And these peptides can be enriched using specific antibodies and quantified using mass spec. And the nice thing about this technology is that you can actually map the exact site of modification. For proximity labeling, we use a turbo ID system, in this case, N-terminally tagged Cerblon, inducibly express this construct and treat the cells with the glue and along with biotin and enrich the biotin-related proteins and quantify them by MS. Just a few words about the selection strategy for the cell lines. So what you see here in this PCA is a proteomic profile, or we profiled these cell lines using proteomics, and you can see that some of them cluster closely together, some others are more distant. Cut a long story short, we ultimately selected two cell lines, namely U7 and NB4, because of their complementary protein expression profile, which you see here in this Venn diagram. But this was not the only reason, because I think if you chose any more or less random pair in this PCA, you will get a similar plot as long as they are distant enough in this PCA plot. But more determining for us was the responsiveness towards Cerblon-based glues. So we treated most of those cell lines with a number of tool compounds and assessed their responsiveness using global proteomics. And what you can see here is comparison of NB4 JURCAT and U7 versus A498. And you can appreciate that these two cell lines have more proteins that are colored in red. These are significantly downregulated proteins. And this made us ultimately choose NB4 and U7 for screening. Interestingly, when we look at the Cerblon expression in those cell lines, so we can see a certain correlation with responsiveness and Cerblon expression. For example, if we compare U7 with A498, we can see that U7 has significantly more Cerblon. However, when we look at, for example, JURCAT versus NB4, we can see that JURCAT has higher Cerblon levels, but according to our analysis, it's less responsive to Cerblon-based glues than NB4. So meaning that the responsiveness to glues is only partially explained by endogenous Cerblon abundance. In this slide here, I summarize what we found. So this is compound activity profile of U7 and NB4. So the take-home message is here that we identified in both cell lines about 50% of compounds that displayed low activity, meaning that only between 1 and 10 proteins were significantly downregulated. And I believe that a good number of those compounds would probably have not manifested any effect in a phenotypic screening assay. And then we looked at the published Neosubstrates that I quoted earlier, and out of those 50 proteins, we detected 25 that were downregulated at least once in one of the two cell lines that is depicted here. And for 17 of those proteins, we could confirm regulation at 6 hours, and we could also showing a rescue when we co-treated with MLN 4924, demonstrating a calendering ligase dependency. Excuse me. Here you can see how we selected Neosubstrates. So these plots show here promiscuous degrader and a number of proteins downregulated when the compound treatment was done for 24 hours. we would follow such compounds up with a shorter treatment time here, 6 hours. And I forgot to mention, just for those among you who are less familiar with these kind of plots, on the x-axis here is the log2 fold change. On the y-axis, we show the standard error. Each dot represents one protein, and red means significantly downmodulated. So you can see that we can recapitulate the regulation seen at 24 hours for some of those proteins, meaning that indicating these are most likely primary substrates. We can also show that their regulation is current ring ligase dependent. Then we would take such candidates and perform our global ubiquitinomics analysis. And in this volcano plot here, you can see that the dots are shifted to the right, meaning these are upregulation events. And each of these dots now represents one ubiquitination site. For example, you see here RAP28 and lysine 181, which was ubiquitinated. To visualize which of the proteins were regulated in proteomics and ubiquitinomics, we can generate such a plot here, where on the x-axis you see the regulation in proteomics, and then the y-axis in ubiquitinomics. And the proteins colored in turquoise here are the ones that score positive in both assays, and we classified those as neosubstrates. Next to such more promiscuous degraders, we find very selective degraders, like shown here for FIS1. And there are a number of those actually that we identified. So when looking at a few more examples, we can see what I already showed in the last slide. So very selective degraders like this one here for KDM4B, but also a more promiscuous one. In case of the latter ones, what we noticed was that there was an aberrant co-degradation of protein complexes, like shown here for CDK7 cyclin H MNAT1, and in this case for CDK13 cyclin H. So what we noticed though was that when you look at the proteomics, you can see that one of the complex partner displays a stronger downregulation compared to the other partners, which in this case is MNAT1, and in this case was CDK13. And we also predicted G-loop for MNAT1, but not for cyclin H or CDK7. When we were about to submit the manuscript, Fisher-Donovan paper came out showing nicely that MNAT1 was enriched in their interactomics assay in both cell lines. And Monterosa predicted a G-loop on MNAT1 and nicely demonstrated abrogation of binding when the glycine was mutated to the asparagine using nanobread. For the CDK13, actually, we were not aware of any molecular glue degrader targeting CDK13. And the reported cyclin K degraders that you might be aware of, they act through a distinct mechanism. So we tried to predict the G-loop, and that's what we found. So this is CDK13, and there is what we think is the G-loop. And we tried to do the same for cyclin K, but did not identify any, indicating that most likely what we are seeing here is a primary glue target. Looking at the biological diversity and how this influences neosubstrate degradation, I show this example here where this is the same compound used for U7 or MB4 cell treatment. And you can see here casein kinase 1 epsilon significantly downregulated in one cell line, but not in the other. And when plotting these two profiles against each other, you can see the proteins labeled in red here that are regulated in both cell lines, but the cell type specific ones for U7 here are blue, and pinkish color are the ones specific for NB4. So interestingly, when we look at the expression of casein kinase 1 epsilon in those two cell lines, there is absolutely no difference, indicating that there is a cell type specific substrate competition. Down here, there is another example differences in the two cell lines. So the KDM4B degrader, a very nice downregulation KDM4B in Hu7. The same is true in NB4, but additionally here we detect two more neosubstrates, VCL and BISL. And when performing global ubiquidinomics, we noticed that we detected VCL ubiquitination in both cell lines, showing that the sensitivity of our ubiquidinomics assay can be higher compared to proteomics, and also that here we are seeing ubiquitination, which leads to a productive degradation and also a non-productive ubiquitination events. And this is something similar to what has been described by Rebecca for the interactors, with the difference that we are a bit more downstream here. When looking globally at our data set and trying to find out how these non-productive ubiquitination events classify, so here is again the plot that I also showed previously with proteomics on the x-axis, ubiquidinomics on the y-axis, and the proteins colored in blue are the ones that are ubiquitinated but not downregulated. And as said, we had a look at all of those and classified them. And interestingly, what we find is that 30% of those are known neosubstrates. Also, that about 20% contain a kinase domain and about 25% contain a C2H2 zinc finger domain. And this is the distribution of domains that we find within these proteins. And if you look at these and compare it to what the Donovan Fisher lab has published for the interactomics, and also what Monte Rosa has found for their predicted GLUE proteins, then we will see that there is actually quite a significant overlap, meaning that a number of those candidates are most likely neosubstrates of cereblon. You just need the right compound to maybe have the right orientation in the complex and trigger a productive degradation. So this is kind of a summary of the primary screen. So we had nominated 28 neosubstrate candidates, and out of those, we were able to validate 17 using ubiquidinomics. For the remaining 11, we noticed that these look quite convincing in terms of regulation, and one of those was G3BP2, which is shown here. So overall, quite selective regulations, but their fold change was mostly very moderate, indicating sensitivity limitations, and also probably due to neosubstrate competition in this background. So we are quite intrigued by a study from the Herbert lab showing that neosubstrate degradation not only depends on the chemistry, on the used compound, but also on the neosubstrate expression and how and the expression levels of competing substrates, and most importantly on the expression of cereblon. And that is what is depicted here. So here we see IXZF3 and CK1 alpha, and you can see the blue bar is a condition where cereblon is overexpressed, and you can detect stronger degradation rates for those two proteins, indicating that cereblon expression nicely correlates with neosubstrate degradation. So we adopted this system, generated a cell line, a hex cell line with cereblon overexpression, and retested those compounds. So here we can see a proteomics profile, so G3BP2 in parental cells, and very moderate downregulation. Upon cereblon overexpression, you can increase this significantly, and you add a number of other proteins that are being downregulated. And if you look at those, you can, the ones that are familiar with cereblon-based glue degrader proteomics, you can recognize a number of those, and many of those are indeed validated cereblon neosubstrates. So this nicely validated our system, and so we performed ubiquidinomics in this context. And what you can see here is that we nicely validated G3BP2 ubiquitination, and also using our TurboID MS platform, we can see that G3BP2 is nicely enriched Also, I think what you can appreciate here is that USP10 is a complex partner of G3BP2, was both ubiquitinated and enriched in turbo DMS, but we suspected G3BP2 to be the primary target because of its stronger enrichment with both methods. And in the meantime, Monte Rosa has actually demonstrated that G3BP2 is indeed the primary target. This is study from a few days ago, I think, or last week. So where you can see G3BP2 tested in nanobread and nicely binds to cereblon, and this is not the case for USP10. And they also disclosed a structure actually showing that G3BP2 engages cereblon in an unconventional way involving the LON domain, and also that this binding is not mediated by a G loop. Staying with the G3BP2 degrader, we detected several downregulation events for this protein, or both proteins actually, G3BP2 and USP10. And some of them look quite messy, like this one here, and others were very selective, like these ones here. And what we noticed was that the more selective ones were actually those compounds that contained a phenylglutaramate binding core for cereblon. You can also see a very steep SAR. The only difference between these compounds is the shift of this fluoride atom from this position to this position, and you get a much stronger downregulation of G3BP2. So looking at this phenomenon in more detail, what you see here is a heat map. The color scale here is a log2-fold change. Up here, we classified the compounds as to whether they were containing a PG core or an IMID-like core. And what you can see is immediately, so many of the known IMID targets like RAP28, ZFP91, or GSPD1 were very frequently hit by these IMID-like compounds and were almost never downregulated by the phenylglutaramate compounds. So, and we also, as shown also here, saw that these phenylglutaramate-based compounds exhibited a much higher selectivity. So as a summary of this first part, I show this plot here where we have plotted the compounds here on the left that regulated the neosubstrates here in the middle, and also on the right-hand side in which cell line they were identified. So in total, we have identified 50 neosubstrates, both detected in proteomics and validated with ubiquitinomics and other technologies in this screen of 100 compounds in 2 cell lines. So that was quite encouraging. So of course, we wanted to see whether we can further expand neosubstrate identification. So we thought of expanding this a little bit. And so we screened 1,000 compounds using a very similar workflow. Similar to what Catherine has shown, we tried to improve both sample preparation throughput as well as mass spec throughput. And we have quite a good throughput now. We screened these 1,000 compounds in about 6 weeks. And the obvious thing to do is look at how many substrates you can identify. But the other nice thing about these large data sets is that you can use machine learning trying to connect compound structure with proteomic profiles. So here is just a QC kind of overview of these larger screens. So you can see very good quantification precision with CVs of below 5%, comprehensive proteome coverage, and very high data completeness. Again, the pie chart as shown previously, and very similar profile actually, about 50% of compounds, again, displaying low activity, which was defined as downregulated only between 1 and 10 proteins. And looking at how many novel neosubstrates we can identify, actually, in this out of these 1,000 compounds, we have 60 potential novel degrader targets. And it's important to mention that for this kind of plot, we exclude all highly active compounds because they would massively inflate these numbers. So we just used the selective ones, and we also excluded all the neosubstrates that were described in the literature and all the ones that we have identified. So to look more into what we can find more if we expand biological diversity, we rescreened those compounds in a second cell line, and again could identify 50 novel degrader targets, indicating that to maximize neosubstrate yield in such screenings, you have to diversify both chemistry and biology. Looking at the what we find, frequently we identify IMID targets, like shown here with many compounds. But I think the take-home message here is that we have many candidates, about 70, which were downregulated at very low frequency, just by 1 or 2 compounds. Looking at disease relevance, you can see that a number of those are disease relevant, not just when looking at cancer, but also for immunology or neurodegenerative diseases. And as said, when having such large data sets at hand, we can now try to connect the compound structure with the proteomic profile, and that is depicted here. So here we wanted to look at fingerprints that determine V1 degradation, and we could identify fingerprint 662, which is depicted here as the strongest contributor for V1 downregulation. And looking at the compound here, you can see that fingerprint 662 is highlighted in yellow, and the proteomic profile, we can nicely validate our predictions, showing about 4-fold downregulation of V1. Can turn this around and try to predict undesired off-target effects. So what is shown here is predicted log2 fold changes for CK1 alpha with or ZFP91, and the 3 colored lines mean indicate compounds with log2 fold change near zero, which we used for follow-up proteomics analysis, and you can see that we could nicely verify our predictions here. So with this, I'm at the end of my talk. I hope I could convince you that global proteomics and ubiquidinomics are a powerful combination for neosubstrate discovery, and that in such screenings, not just the biological context, but also the cerebral expression level matter, that ubiquidinomics can capture both productive and non-productive ubiquitination events, and that the chemical and biological diversity or the combination of those two can maximize the yield of new substrate candidates identified in such screens. And because proteomics is now very high throughput and very reproducible, it enables screening of very large compound libraries, and it can actually be used as a first-line screening tool for such degrader libraries. And when doing so, you get large data sets, which nowadays can be used for predictions of compound selectivity, which I showed a proof of concept just a few slides ago. And if the data sets are large enough, these can support rational design strategies for glues in the future. With this, I would like to thank the entire team at NEOsphere, all of our collaboration partners, especially Zoran and Giselle from St. Jude, and all of you for your attention.